Hello, my friend. Today we talk about neoplasia and cancer. And what is the relation between neoplasia and cancer? We will discuss in this video. And let us define what is neoplasia. So neo means new. Neo means new. This you know already. Plasia. What is plasia? Plasia means growth. So neoplasia means new growth. What is that new growth? I'm talking about the abnormal mass of tissue grow in the body. What is that? Abnormal mass of tissue grow in the body. So that's we call neoplasia. Okay, but by old definition, they just define that neoplasia means new growth, but that is not clear. And what is cancer? To understand cancer, then you must understand like this. For example, I have a cell. Okay, I have a cell. So I'm talking about the human cell. I'm not talking about the bacteria cell. Okay, and in human cell, what we have? We have in the center, we have the nucleus. We have nucleus. Okay, bacteria cell don't have nucleus, but human cell have nucleus. And within the nucleus, what we have? Let's suppose this is the nucleus. Yes, this is the nucleus. And inside, yes, inside this nucleus, what we have here? We have chromosome. Here we have chromosome. Let me write out here. Chromosome. Chromosome is a genetic material from the parents. Clear? And then this chromosome, within this, we have DNA. What we have? DNA. And what is DNA? Let me define very simple. DNA is just the deposition of the nucleotide. For example, I have a piece of DNA here. Then you will see like this. DNA is a deposition of nucleotide. Deposition, double means two strands of nucleotide. And what happened on this strand of nucleotide here? We have genes. So what we have? We have genes. Okay. And what happened in cancer? In cancer, if somebody has cancer, and what happened? The genes are mutated. The genes. For example, this is the gene, and the genes are mutated. The gene on the DNA are mutated. Mutated. So how do you define mutated? If any changes in the DNA, so that's we don't call mutated. So when the genes on the DNA mutated, it means it changes the genetic program. Clear? Again, the genes are changed. The genes change the genetic program on DNA. So that's we call mutated. And then it has ability to inherit. You know, inherit means from mother cell we call original cell. And when cell divide, from one become two cells. And this mutated gene sent to this mutated genes transfer to daughter cell. This is original cell, and then we call daughter cell. When the cell divide, one become two, two become four, like that. So they carry what? They carry mutated genes, and they send to the daughter cell, or you can call this baby cell. And from one cell become two cell become four cell, like this. And what they carry inside? They carry the mutated gene. So that we call cancer. From one original cell develop or multiply, become thousands of cells. And on that cell carry what? This gene, mutated gene. So we call cancer. That's in the simple definition of cancer. But now we go into detail. And let's look at how the normal cell can turn into the cancer cell. Okay. You know why we got cancer cell? Why everybody got cancer cell? Due to some reason. Okay, due to some reason. Number one is by what? Radiation. By radiation. Okay, so by radiation we can get the cancer cell. Clear? Second, by what? Oncogenic viruses. By oncogenic viruses means some virus can bring the cancer cell or can make the cancer cell. Yeah. And the third reason by chemical, by chemical carcinogenic substances. So what does it mean? Means by some chemical or substances, and that substances can produce cancer. We call that carcinogenesis, means give cancer. Yeah, the substance can give the cancer or can turn the normal cell into cancer cell. And fourth, number 4, by mutated genes inherited. I said mutated gene. You understand already? Mutated genes again. I repeat that. Mutated genes are the genes, already changed the genetic program. And then they can transfer to the daughter cell. From original cell can transfer to the daughter cell. And they multiply. They multiply until they become a max, a lot of. Then inheritance means from the parents, and then they inherit to the children. Means these genes, mutated genes from the parent, now they transfer to the children. So this is the 4 basic reason, and that one person they got the cancer cell. Okay, now let's look at the detail inside the genes of normal cell and cancer cell. Now I divide this into two columns like this. And on this column, this side, I will draw simple diagram of the normal cell. You see, normal cell. And here, cancer cell. And now look at the difference. What is the difference? By simple diagram, then I will draw like this. Simple diagram, I will draw like this. Okay, now on the cell, this is one, for example, this is the one cell. And I have some sets of genes here. The cell has some sets of genes here. And what is that? First set of genes, this gene we call proto-oncogene. Proto-oncogenes. Okay, proto-oncogenes. And here, another set of genes. This gene we call DNA replication. And here, we have another set of genes. This gene we call tumor suppressor gene. Tumor suppressor genes. Okay. Now basically we have three in the cell. We have these three sets of genes. These three sets of genes are what they are doing. Okay, in the normal cell, these three sets of genes, they are working very strictly. Okay, so when they, okay, just I will draw here the same for cancer cell. We also have the sets of genes here, but this is the cancer cell. These genes are mutated, and then how they are mutated. And let's see. So normally, first proton coachings, they just work when they receive the signal from the body. For example, here, if you have, if I have, this is the piece of the skin here. This is the piece of the skin here, and then what happens if this skin have injury here? If you have injury here, then what happens? Then the body will send a signal. Now they start to work, but how? They will send a signal. The body will send a signal. The body will send a signal to these genes, proton coachings. And what they are doing? This gene proton coaching will send a factor. Now they will send a factor, and this factor will stimulate this DNA replication. So it means now the gene this gene will work on DNA replication. Now the DNA start to replicate to make the new cell, to grow the new cell. For what? To fix, for example, to fix this injury. To fix this injury, so we need new cell. Okay, we need new cell. And these genes, proton coaching, receive the signal from the body, and they stimulate this DNA replication. And then when these genes DNA replication, they complete the work. Okay, the work has been done, and then they will receive. And how they stop working? Okay, when they complete the work. Okay, for example, they complete the work, then they are tired, and then they will receive from here, from the cell. We have another gene. This gene we call tumor suppressor genes. This gene will start to work. They will send a signal to these genes, and this gene to talk to these genes stop working. Okay, so they work very, they work very strictly like this. This gene will start release the factor, and they start to work. This gene talk to this. This gene talk to this gene, start to work, making the new cell. And when enough cell, for example, when they already making the new cell enough to fix this injury, and then when they reach the target, and then this gene will tell to this gene stop working. Okay, this is the how normal cell they are working like that. And what happened with the cancer cell? See, cancer cell, this gene proton coaching, so they are mutated. They can, by itself, they can promote this cell DNA replication. They can promote this cell to start to work, to replicate the DNA, to make the new cell. Okay, so they don't need any signal. They have ability to work on itself. It means, for example, this citizen, they don't obey the law of the government. So they are autonomous. They can make their new law. For example, this citizen, they can make their law, and they promote this, they stimulate or promote these genes, DNA replication, start to work. Okay, now this gene will start to work and making the new cell, like this. Okay, and then these genes, tumor suppressor genes. So these genes suppress the DNA replication. Suppress means stop the DNA replication. When they send the signal, and this gene have to stop, but when this gene in the cancer cell, these genes they lost their function. Okay, they cannot send the signal to here. They cannot send the signal to this this gene. So these genes continue to they obey. This gene obey this gene. They continue to work to making the what new cell, to making the new cell like this. But they don't stop. Okay, this is the second characteristics of the cancer cell. First, this gene is autonomous. They have a capacity to work on themselves. Okay, they don't obey the law of the body. Okay, it means these genes are mutated. Clear? And these genes means now they gain one function. Okay, they have ability to promote these genes work, but here they lost the function. Now even they send a signal and they don't obey. Okay, but remember one thing: if any changes in the DNA, then we cannot say that is mutated. We cannot say mutated. Why? In the normal cell, if these genes are in the normal cell, if these genes are changes or damage, we call that damage. And then what happened? Or if even this gene, tumor suppressor genes are not mutated, that is damage. And then what we have here? In cell, they are very intelligent, so they are repairing everything here. Means now they have a set of gene here. They have another set of gene. It is like a tool. Okay, and this tool we call DNA repair. DNA repair genes. So if the genes, some genes in the cells are damaged, okay, and don't worry, we have the DNA repair genes. So these DNA repair genes will fix the genes are damaged. So don't worry, every day in our body, thousands of genes are damaged. And then we have the tools, the cell have the tool DNA repair damaged genes. Okay, so don't worry about that. This is the normal cell, it's work like that. But even, for example, if these genes are damaged also, if these gene DNA repair genes are damaged and they lost the function, then our cell is very intelligent. They repair another gene also. Now they repair another genes. We have another set of genes on the normal cell. What is that? What is that? We have one pair, okay, of genes. One pair of genes. Okay, and let's go. We have one pair of genes here. So what is these genes? These genes, they works as a pair. The name of this gene is apoptosis genes. Anti-apoptosis genes. What is apoptosis? Apoptosis means the process of forcing the cell to die. So what happened if apoptosis means once genes are damaged and DNA repair genes cannot repair, and even DNA repair genes are lost their function. Okay, they cannot work. Something wrong with them. What happened if something wrong with the genes, something wrong with the gene, or something wrong with the genes, and then they start to work. But if there's something wrong with the genes also, now we have another tool here. The gene have another tool. These genes, okay, this gene we call apoptosis genes. Apoptosis gene. So normally this apoptosis genes, they are sleeping. They don't work. They don't act. They don't they don't they don't work. They are sleeping. Okay, But when these genes have problem, and then they start to wake up and start to work, and what they do, they will force the cell to die. Yeah, they will force the cell to suicide. To suicide means going to the process of apoptosis, means the cell will start to die. For what? To prevent cancer. If the cell, if the genes are mutated or damaged, and it cannot be repaired, and it continue to survive, continue to live. Okay, and that cell become very dangerous cell. But it's okay. We have these genes. These genes bro apoptosis genes. They wake up. Okay, then they on, means they on. They start to work and they will kill the cell. Like that. These genes will kill the cell, suicide. So to protect the body from the cancer. What happened? And how about this in anti apoptosis, means this is against the apoptosis. These genes, anti-apoptosis genes, they against apoptosis, means they don't allow to the cell to die. What happened? These genes also very important, because the lifespan of the genes, how long the genes or the cell can live or can survive, is depends on these genes. Okay, for example, some cells are living some days, few days, some cells living 1 year, like that. So depends on these genes. So normally these genes, okay, is working, active, okay, and these genes are sleeping. But if if this gene is on and this gene will off, this gene will be off. This gene, when this gene is on and this gene is off and on. If on here, if this anti-apoptosis gene is on and this gene will off, like that. Yeah. So this gene very important gene. So they will kill the cell. Yeah. And we are safe. If this gene is working, then we are safe. The problem here is on the cancer cell. So on the cancer cell here, this gene is working, anti-apoptosis gene is working. They gain the function. But this gene is not working. So when DNA repair genes are not working, and they don't active, they don't work also. Okay, in the cancer cell, these genes cannot work, means they lost the function. Now again, you see in the cancer cell, you see here these genes gain the function. They gain the function. Okay, means they overwork, and these genes also gain the function. They also overwork, but these genes lost the function. Okay, they cannot work. They lost the function. And also, these genes also gain the function. Means now the genes are mutated, and the cell continue to live. The cell continue to survive. Okay, but these genes cannot kill the cell. So here they gain the function. Here they lost the function. You see? So this is the basic understanding about the basic difference. Okay, I'm talking about basic difference between cancer cell and normal cell, and how they work like this depend on the genes like this. Okay, so in the next video I continue to share with you next step. If these genes are damaged, okay, the cell continue to and this gene is working, anti-apoptosis genes are working, and these genes are cannot working. Okay, something wrong with them already, and these genes continue to work, and we have another genes also, and it's really from the normal cell turning to the cancer cell. Actually, it is not easy. We also have another set of genes working in this also. So I will share with you in the next video. Thank you so much.